So now let's hear from the other end of the spectrum. Walter Ogier, President and CEO of Acetylon Pharmaceuticals. Acetylon is an early-stage innovative biopharma startup. They're working on isoform selective HDAC inhibitors based on technology developed at Dana-Farber and the Broad Institute. Their $40 million in fundraising over the past few years, including a $27 million Series B announced just today, in BioWorld today, will fund them through a phase 1-2 clinical trial of their lead candidate. And it's all come principally from individual private investors, augmented by funds from a nonprofit foundation. They haven't tapped VC at all. Walter, maybe you can tell us a little bit about how you decided to take a non-VC route and how you found these alternative investors.
I'm happy to. Thank you, Lynn. Maybe a couple words of my background, and then I'll get into talking about the company and the financing. I'm the non-expert in this company. While I have a background in organic chemistry, we are a small molecule company. I've had experience in developing cell therapy companies, including working with Amgen to build a 40,000 patient stem cell transplant franchise, i.e. bone marrow transplants and peripheral blood stem cells, while I was with Baxter for almost a decade. This is my fourth time as CEO of a company over the past 15 years. I had a successful exit in there. I've worked on monoclonal antibodies, gene therapy, a company I started, or actually was CEO of it, already been started, but is now called Bluebird. It actually appears to have potentially cured a patient with thalassemia major in Paris, and it was announced in Science a couple months ago. And I've raised about $150 million of money in my career, most of that venture capital, a little bit of hedge fund money as well. I'm CEO of private and public companies. So I didn't have any bias against venture capital when I came into this company and helped start it. But if I go to the next slide, we can talk about some of the history. As Lynn mentioned, we are a spin-out from the Dana-Farber Cancer Institute and Harvard's Broad Institute. Ken Anderson, one of the scientific founders, is one of the leading clinicians in multiple myeloma in the world today. He's been the lead investigator on essentially every important new drug approval in multiple myeloma over the past 15 to 20 years. So he had put together a nucleus of our research effort between the Dana-Farber Cancer Institute and the Broad Institute at Harvard University, and was really the catalyst for getting the company founded in October 2008. We are focused on isoform selective HDAC inhibitors. This comes via acetylase inhibitors. There are two drugs in this class on the market. So Lynn's has been on the market for 4 years now. Istidac's just coming to market this year. And there are numerous others in clinical development. They're all of the category of what we would call pan-HDAC inhibitors. They're not isoform selective. There are actually 18 different HDAC enzymes present in human cells. And these drugs inhibit a fair number of those. And most of them were discovered and developed back when they didn't know all of those different isoforms. They all have therapeutic effectiveness. There's been really no controversy there. But they do have substantial side effect profiles. Because they're not specific, they tend to have side effects that are actually seen with other cancer drugs, which limits their ability to be put in combination therapies for other cancer with other cancer drugs. The way of treating cancer today, in almost all indications, is combination therapy. So these side effect profiles are really quite critical. If you have overlapping side effects, you can end up with patients that will only get through 1 or 2 courses or cycles of therapy and then have to come off the treatment and try something else. So at the Dana-Farber and at Harvard, they had put together a really compelling discovery platform based on technology that we term iterative biasing chemistry. This work has mostly been done in the academic setting. We had rights to it and doing a little bit of it, but also had tried to follow, particularly early on, a virtual model, much like the one you just described. We're also working on second-generation drugs, so the risk here is somewhat lower than going into an entirely new class of drugs. I think that was helpful in getting the company founded and also helpful in coming up with lead compounds for us to begin our activities. So we were given probe compounds, and in fact a nice lead molecule for HDAC6 inhibition, and I'll talk more about that in a second. HDAC6 inhibition had already been shown in the literature and in research that had been done on our two founding institutions to provide a lot of the advantages that one would need to get rid of the issues of side effects with this class of drugs, and provide potential enhanced therapeutic outcomes, particularly via synergy with other drugs in the cancer area for leukemias, lymphomas, and particularly multiple myeloma, where we are staking our first bet in the clinic, and a lot of promise for other diseases as well. Inflammation, autoimmune diseases, for example, where chronic therapy would prohibit any of the existing drugs in the market from being used in any kind of long-term therapy. So let's go to the second slide, or the third slide. So this gives a history of our company's operations and financial progress. The company was founded again in 2008 with several $100,000 inputs of seed financing from a group of first private investors, ultimately 3 individuals who have really formed the nucleus around a lot of our fundraising. These are multi-millionaire folks who had some passion for the disease, had some connection with these institutions, the academic institutions, and came together to provide the funding to start the company, to bring me into the picture. I had already done a Dana-Farber company earlier and had a good exit there, so I was a known quantity, was asked to come in and be the proto-CEO for this company. But like a lot of companies that are getting started today, I was working half-time out of my family room, looking at the backyard and talking a lot on the phone and working on computer presentations. We were virtual operations for the first year. We spent that year negotiating a technology license with Harvard and Dana-Farber. With 2 difficult parties, that took quite some time, but we have a good deal at the end of it. We started our lead optimization work in Asia based on a relationship that was already existent with the Broad Institute, the CRO in Shanghai. This has become, I think, a dominant model for medicinal chemistry. It seems to me that a lot of medicinal chemistry is going overseas these days because of the cost advantages. All of the managers in that CRO speak English, have worked in the United States. It feels very much like the kind of relationship that you heard about just now for clinical development, working virtually. We were able, not quite a year later, to raise a $7 million Series A preferred round. And this involved these initial seed investors plus various friends and contacts of theirs. The fundraising that we've been able to do has largely been done by networking. It turns out that people who have $100 million pretty much know each other in this country. And if they're passionate about something, they are able to bring their friends into financing deals. We brought a further investor in a $2 million convertible note. This was done as a note about 5 months after the initial Series A that would convert into the Series B, because by that time we already had a lead compound in hand and a candidate that we were about to nominate for clinical or preclinical development. The feeling was among the investor group that the company had been substantially de-risked at that point. Maybe I'll go back and just say, at the beginning, as we were looking at going down this path, we did talk to VCs to some extent, but there was a feeling around the table that the investors who were the seed investors were a little bit afraid of venture capital, maybe for good reason, and decided that they would like to try to do it among friends of theirs rather than bringing in folks that might have ulterior, very different motives in terms of where they want to take it. I think there was also a great passion here for seeing new medicines reach the market, so notions of a purely financial motives around the table were a little bit suspect. And in that setting, it became fairly straightforward to get the board of directors and these seed investors behind us for raising money privately, and that's where we've gone. In fact, so earlier this year we were able to work to bring in funding from the Leukemia and Lymphoma Society. This is a nonprofit group, like others out there, a patient advocacy group that's been successful in their own right in raising funding, philanthropic funding from a number of donors. They've funded, I think, somewhere in the neighborhood of 20 to 30 different projects and companies, and they don't have a resistance against doing it with private companies, not just academic settings. And they set us up, and we'll get a sort of milestone tranche investment from them over the course of our phase 1/2 clinical trial of multiple myeloma that will total $4.9 million.